question 14, and that's Miss Dixon's side. Which of the following options A to D is the stage of mitosis shown above? So you should know IPMAT, I pick my apple tree. So in interphase, which you learn about in GCSE as well, that's when your DNA doubles amongst other things. Prophase, that's when we get, sorry, this pen's really annoying. Prophase is when we get the DNA supercoils and condenses and shortens. And other things will happen, like the centriole will move to the two separate ends and the spindle starts to form. Metaphase, so let's put that in actually, spindle begins to form. Metaphase is where they line up in their little pairs down the middle. Anaphase is when the centromere, which holds each of the pairs of chromatids together, splits. And then telophase or telophase is where they reach the opposite ends. Kind of like so. Okay, so this one has got them lined up down the middle, so that's metaphase, the one in the middle. Metaphase, it's the middle thing that happens if you include interphase, and it's where they line up down the middle, M for middle, M for metaphase. Okay, so that should be B. Okay, next one, antibody. So this is my stuff. You should be looking and you should be thinking before you look at the question again. A, right, that's our variable region, which has a specific shape, which is going to bind to our antigens on the surface of our pathogens. B, this is our disulfide bridge holding it together. C is our hinge region. So we can, I'll just leave it like that actually, which allows it to bend so it can stretch out. And D is pointing to our heavy and light chains, or just polypeptide chains, I suppose. Which of the letters A to D identifies the region of the antibody that allows the distance between the antibody binding sites to vary? So the variable region, here and here, that's our antibody binding site. So if you just look at the structure, how can we vary the distance between these two bits? It must be these bits here. Imagine it's like a bendy straw. That bendy bit on the straw is going to allow it to bend like that. Okay, so it must be the hinge region, C. Next one. A group of students were investigating the diffusion of molecules across membranes using a model cell as shown below. Now this is Miss Dixon, but I think this is kind of GCSE knowledge actually. Quite a high, like hard GCSE question, but because there's quite a few elements to it. Biochemical tests, were used to identify the types of molecules present. The results are shown in the table below. A tick represents a positive result. Which of the rows A to D shows the correct results for the cytoplasm at the beginning of the experiment and the extracellular fluid at the end of the experiment? So we've got Benedict's, Biuret and iodine. So you should be thinking, right, Benedict's, that tests for sugars. Biuret is proteins. and iodine is starch. So again, I'm not going to look at the options because that's just going to confuse me. It's a bit overwhelming when you look at it like that. So we've got our model cell. Here's our like viscing tubing, which means we know that it's partially permeable. We've got our extracellular fluid here. So at the start, we're going to have inside our little cell, we've got starch, we've got amylase, and that's it. So this is for inside the cell. So we would expect to have a positive result for our iodine test. And amylase is an enzyme. Is it still filming? Yeah. Amylase is an enzyme, and enzymes are proteins. So we would expect to get a positive result for the Biuret test. Okay, again, I'm not going to look down here just yet. At the end, inside the cell still, we would expect our starch to have been broken down, and some of that's going to have moved out as well. So we're still going to have our enzyme in there. That's going to be too large to move out. So we're still going to have amylase, which means we'll still get a positive Biuret result. And our starch should have been broken down. So we shouldn't really see a positive iodine. Assuming it's all completely broken down. But we'll come back to that bit. Like we do on anything, if we're a little bit unsure, it should hopefully make sense with our options. If completely broken down. If this was like a long answer question, you could say that. You could say, I would expect to have a negative result for iodine if it's been left for long enough for it to be the starch to be completely broken down. What is starch broken down into? Glucose. So we're going to expect to have glucose in our end product. Which is tested for with Benedict's, so we would expect a positive result for Benedict's, assuming it's not all diffused out. Outside the cell, so the extracellular fluid, outside cell. In the beginning, now does it tell us anything? Just come back to it. Which of the rows shows the results for the cytoplasm at the beginning? Okay, so we only need this for the beginning, and we need the final, what we're going to see in the outside of the cell. Okay, so assuming we've got none of these in the beginning, at the end, so nothing, in the end, we're going to expect to have seen our glucose molecules diffuse out, so we should get a positive Benedict's. Amylase enzyme is going to be too large. That's still going to be inside our cell, but it's going to be too large for it to come out. So we're going to expect a negative amylase. No amylase, so we should expect a negative Biuret test. And starch is a large molecule, so we wouldn't expect that to be able to diffuse out either. So starch, which is our iodine test, we would not expect to see. So again, come back to the question. Used to identify types of molecules present, the results are shown in the table below. Each represents a positive result. which of the rows shows the correct results for the cytoplasm at the beginning? That's what we've got here. And the extracellular fluid at the end of the experiment. That's what we've got here. So our cytoplasm, we want to see positive iodine, positive biuret. So cytoplasm, positive biuret, so it could be any of these, not this one, and positive iodine. So, well, that doesn't narrow it down on that one. We would not expect to see any positive result for sugars because it's right at the very start, so the starch hasn't been broken down yet, so we would be expecting to see this one. Okay, but I'm not going to cross the others out yet, just in case. In our extracellular fluid, we've said we're going to expect to see the Benedict's solution showing a positive result. So Benedict's, any of these three, not this one. We wouldn't expect to see anything else on this one. So it's not going to be this one. Oh, sorry. It could be this one. It won't be this one. Could be this one, could be this one. Could be this one, could be this one. It won't be that one. Could be this one. So now I'm going to see which one's got my little circles in every single one. A, yes, 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 yes. So I think it's A. I'm just going to check. Yes, 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 no. Yes, yes. So not B, because this one's wrong. We would not expect to see a positive protein test because the enzyme's not going to be able to get across the partially permeable membrane. C, we would not expect to see sugars inside the viscing tubing right at the start because the starch hasn't been broken down yet. We should see protein because we've got our enzyme, and we should not see, hang on, in the extracellular fluid, we would not see starch because it's too large to cross the partially permeable membrane. So definitely not C. D, that's wrong, that's wrong. The rest are correct. So we shouldn't see sugars inside of here at the start. We should see sugars outside of here at the end. So this one is definitely wrong. So like I say, that question, there's a lot to it and lots of little bits, but actually the knowledge that you have to have is not too tricky, but I think it's the application and understanding the wording. So that's a good one for us to get in practice, I think. Okay, do a couple more on this video. Which of the cells below, represented by cubes A to D, has a surface area to volume ratio of 3 to 1? So this is a question on my stuff. Okay, so this one here is 1 micrometer by 1 micrometer, 2 by 2, 4 by 4, 6 by 6. So let's work out quickly, before my battery runs out, let's work out the surface area of, let's go for the 2 in the middle. So surface area to volume ratio for this one. Surface area will be 2 times 2 to get the surface area of one face, and then times it by 6 because a cube has 6 faces, like a dice. Okay, so 2 times 2 is 4, times by 6 is 24 micrometers squared. The volume will be 2 times 2 times 2. So 2 times 2 is 4, times 2 is 8. 8 micrometers cubed. So that one is 24 to 8, which we can simplify. How many 8s in 24? 3 to 1. Oh, that's what we're trying to work out. So let's just double check. Let's say you've finished your paper and you're just coming back to check things. That was quite lucky, but let's just check this one. Surface area will be 4 times 4 to get one surface, times 6. So 4 times 4 is 16. 16 times 6 is 96. And 4 times 4 times 4, 4 times 4 is 16, times 4 is 56. So 96 to 56. Divide both by 8. So that's 12. And that is, what am I dividing it by? 8. 8, 16, 24, 32, 40, 48, 56, so 7. That took me lots longer than it should have done. So 12 to 7, I feel like that could go a bit more. Maybe not. Okay. So 3 to 1. So this one is 3 to 1. So we're going to go with B. I feel like I've done something wrong here. 6, 45, 46, 47, 48. I'm just halving it. 25, 28. Half again. 48, 24. And 14. Half again. 12 and 7. Oh, okay then. Just doubting myself. Okay. If you wanted to have a bit more practice, you could have a go at this one. Maybe pause your video now before I do it. So the surface area would be 6 times 6 to get one face, which is 36, times by 6 because we have 6 faces. So 6 times 3 is 18, so 180 plus 36 is 216, hopefully. And the volume will be 6 times 6 times 6. Okay, so 6 times 6 is 36. Oh, we've just done that one. That's weird. So it's 216 again. Cubed this time. So 216 to 216 is the same as 1 to 1. And then we would expect to see the smallest one. Well, let's do it quickly. 1 by 1, so we do 1 times 1 to get one surface. Times by 6 to get all 6 surfaces. The volume will be 1 times 1 times 1, which is 1. Don't forget units. So that's our surface area because it's squared. That's our volume because it's cubed. So we've now got 6 to 1, which is right because we should expect to see as an object gets bigger or an organism gets bigger, the surface area to volume ratio gets smaller. The surface area compared to its volume gets smaller. I still feel like I've done something wrong there. I'm sure I'll let you know in the next video. A diagram of a stained blood smear observed under a light microscope is shown below. Oh, I like this one. I bet you lot don't. Oh, this blooming pen. Okay, so we've got different blood cells here. Which of the structures labelled A to D in the diagram is a neutrophil? Right, your neutrophil will be multi-lobed. So it has something like this. Okay, so we can see one here. That might be a neutrophil as well, probably. But we know that B will be a neutrophil. Now I'm just going to label all of them because although I don't need to for the question, it's good for you for your revision. Okay, that's a neutrophil. It's multi-lobed. C, this one is going to be a lymphocyte. Okay, it's the smallest type of white blood cell. and it has a very round nucleus which almost fills the whole cell. Round nucleus almost fills the whole cell. So that's a lymphocyte. Down here, this is going to be a monocyte because it's got this kidney bean shaped nucleus. We'd also expect it to be bigger than a neutrophil, but that's what you would expect to see. But the kidney bean shape is what you want to look out for. So that is a monocyte. Kidney bean, kidney shaped nucleus. And it's the largest white blood cell. Now a macrophage we know is bigger, but macrophages don't travel in the blood, and this is a blood smear. If it said it was taken from swelling from site of infection, then we would expect to see some macrophages as well. But remember that monocytes travel in the blood. When they get to the site of infection, they finish differentiating into macrophages, so they will become bigger. They're too big to actually travel in the blood and squeeze through capillaries. Okay. D, hopefully is obvious, that's a red blood cell or an erythrocyte. Erythrocyte. White blood, red blood cell, obviously. And then here, these little guys, they're going to be platelets, because platelets are fragments of cells. Okay, and they cause the clotting. This one, like I say, I don't think it's kidney shaped. I think it's probably multi-lobed. That one's a bit odd. So which of the structures is a neutrophil? B, definitely. Which of the following is a primary defense mechanism against pathogens? Now then, I've seen this question before, and a lot of students get it wrong because they confuse their terminology, okay? We have our primary defenses, which try to prevent entry of pathogens. Okay, and that's it is non-specific, but then we get our secondary defenses, where we have non-specific, which comes in first, and we have specific. Okay, so primary defenses include things like your skin, your tears, pH, hairs, cilia, stomach acid, because it's still trying to prevent it from getting into your bloodstream. Blood clotting to try and restore the skin, which is the physical barrier. Secondary defense is non-specific. We do have some phagocytosis, non-specific phagocytosis. Inflammation from histamines. So secondary defenses happen once it's inside. Specific is only once the correct antibodies are found, and that's obviously going to speed up phagocytosis, okay, because they can be more specific, and agglutination can occur where it grabs and pulls them all together so that more get engulfed in one go. Okay, so it asks which of the following is a primary defense mechanism, so preventing entry. Neutralization, well, that's when white blood cells produce antitoxins to neutralize the pathogens' toxins. So it's already inside, and that doesn't actually destroy the pathogen at all. That will just reduce symptoms. But it's already inside, so that would be a secondary non-specific. Agglutination, I've just mentioned, that would be specific because it involves antibodies, sticking them together let's have bacteria. Okay, sticking them together so that phagocytosis can be more effective. Phagocytosis we know is engulfing, and it can be non-specific and specific. Blood clotting, we said that restores the physical barrier. So that is trying to prevent any further pathogens from getting in. So it's not neutralization, it's not agglutination because that's secondary non-specific. Neutralization, secondary non-specific as well. Phagocytosis is secondary and it's both non and specific. And blood clotting is a primary defense to prevent entry. Okay, so that would be D. Right, I'm gonna end that video there. Just get that in the middle for you.